So, Scrubbing, I had a request yesterday for some references about the subject. So the problem with like physics, there's not big classes that sell lots of books. So no.
Can you try to be close to the microphone? Even in the Zoom sometimes
Okay, but we wiped out the presentation. Okay, yes. So these are best suggestions I have. So for the molecular biophysics part, this book by Nelson is probably the one that is most oriented towards interdisciplinary students. That is, it speaks physics and chemistry as well as biology students. Biological physics. Nelson is. Can you hear me? Can you hear me now? Is that any better? Okay. So Nelson is he wrote this book from the perspective of neurobiology and the nervous system signals, physiology, but it has some biophysics in it. This is a physical biochemistry. This is more physical chemistry, so it's more molecular. It's more like what we're talking about. It's a big old physical biology of the cell. It's much newer text, and it's a Pearson text. It's in the style of the popular main texts these days, with lots of pictures and things like that. Then for part two, when we talk about systems biology, my best recommendation for the introduction is mathematical modeling and systems biology. Although an introduction of this because it doesn't assume any previous knowledge. It only assumes calculus pretty much, and it builds up the subject. Sorry, and my lecture will actually follow this. There is a newer text out by Nelson that's saying it's biological physics, physical modeling of living systems, and it's less restricted to systems biology as such. It's more it uses methods of systems biology, but it also is concerned with modeling of biological systems from a physical point of view in a more general sense. And I'm actually working on a book myself. This book will be on molecular biophysics, but specifically for nucleic acids. I call it the free energy landscape of DNA and RNA. It's perturbation by ligands. That's the current title, and these are the topics. So first, this introduction and basics of nucleic acids, their composition and their structural properties, and then introduction to the free energy landscape. So looking at the conformation and the energetics of the conformation, building up starting from the individual subunits, the nucleotides, the dinucleotides, then looking at double helices, and then on to RNA, very large scale folding. And then the last part are the interaction between ligands. So that starts with very small ligands, just with ions and salts and water. And then for the zinc equilibrium, which is where we stop. lecture yesterday. And then talks about interactions with small molecules and blood-like molecules, and then interactions with nucleic acids, very important for biology, proteins, and then complex assembly where we have lots of nucleic acids and lots of proteins all together. And then higher constantly, which is the modern studies. And so, like I say, I'm working on this book, but I'm looking for students to read some of the draft chapters and give me some comments. I want to see how it reads for a student. So if you're interested in that, then please send me a message and we can organize, maybe you can volunteer to read a chapter, a draft chapter too. Okay, so remember where we were last time. We've been talking about the importance of the folding of biomolecules, proteins, and nucleic acids. How that's the secret to the great variety of functions of these biomolecules, and in particular, the way they allow the living system to respond to the environment, to interact. And we concluded that what's really important is not just the way it folds, but how the folding changes when it binds to another molecule. And so we looked at different models. There are these kind of cartoon models that describe what happens when the two biomolecules interact, like the key fitting to the lock. Or we talked about that induced fit, remember, where the active site changes its shape, ligand binds. Or the conformational capture, where the active site is dynamic and it's sampling different structures, and when it samples the right structure, the ligand jumps in, fixes it there. And then we said, well, we don't have to just rely on those cartoon models. We could have a quantitative model. And length equilibrium is a very simple concept. The idea is we have two equilibria. One is a conformation. So we have, this is the simplest possible case. A protein may even have two different states, two different shapes. The T square one, the R circular. Then there is an equilibrium between those two conformations. We can define an equilibrium constant L, which is the ratio of the equilibrium concentration at the T state and the equilibrium concentration at the R state. And that's a thermodynamic property of the system. But we have a second equilibrium, that is the binding equilibrium. So our protein or nucleic acid can bind to a molecule. Let's say, for example, a popular early example of this was hemoglobin. Hemoglobin, it's in the blood and it carries oxygen around with the body, so it binds to oxygen. So let's say we have an oxygen combined with R state, and there's an equilibrium constant for that binding. So at equilibrium, the equilibrium constant will be the ratio of the population of the unbound to the population of the bound. And there's also the possibility that it can bind to the T state. So we don't have to assume as in conformation capture. There's no reason to assume that the ligand just binds to one structure. In fact, by default, we would expect it to bind to the different structures, but the affinity will be different. And so the binding equilibrium in the T state will be different than it is in the R state. So we call the binding equilibrium with R state case of R, the binding equilibrium with the T state case of T. And we'll call the ratio between those two C. So if, let's say, the oxygen has a higher affinity for the R state than for the T state, And for the R shape, this number on the bottom will be bigger. So the A will be smaller. And C will be less than 1. And that means when we link these two equilibria, when we add the oxygen, we expect the conformation of equilibrium to move from the T state to the R state. So then we can apply this model in a very quantitative way. Let's say, for example, again, you take that as an example. Hemoglobin combine more than one oxygen. They combine 4 oxygens. So each hemoglobin has 4 binding sites. Now, if L0 was our original conformation, then when we bind 1 oxygen, then we have a new equilibrium constant, which is equal to the original conformation of equilibrium times C. Remember, C is the ratio of the 2 binding equilibrium. And then, if I bind the second oxygen, my conformation of equilibrium changes by another factor of C. Every time I bind an oxygen, the equilibrium constant changes by a factor of C. Now remember, the equilibrium constant is the ratio of the conformations of the 2 states. And that ratio is determined by what in terms of energy? It's determined by the free energy, right? Through the Boltzmann factor, right? So those equilibrium constants are proportional to the Boltzmann factor, which governs this probability. So, let's think about this then, in terms of what it means to the conformation. And in order to do so, we will go back and we will consider at a more fundamental level how the free energy is determining the conformation. And we can visualize that in what we call the conformational states. So if we imagine a plot and between horizontal dimensions, we have the different possible conformations for our bio. Ordinarily, if we were looking at a small molecule, there wouldn't be very many conformations. If we were looking at the state of a system that's a periodic crystal, or let's say a phase like gas, something that converts gas, liquid, solid, there's limited numbers of states in the system. But for our biomolecules, our wonderful Raphael or master-like molecules, as Schrodinger described them, there's a huge number of possible conformations. So we can imagine plotting the free energy, because each conformation has its own free energy. So if we have put the free energy on the vertical dimension, then we have the surface we call free energy landscape, or the FEL. And this free energy landscape will determine the conformation of the biology. So the low regions, the valleys, they're going to be the most popular, right? According to this expression. On the other hand, if the temperature is high, then there's enough ambient energy to jump over these hills which are aligned, and then go to one of the other low energy conformations. So the size of this hill determines what we call the activation limit, which you might remember from your kinetics, your chemistry. So now let's combine this idea with a linked equilibrium, because the equilibrium constants we talked about, they're just proportional to the Boltzmann factor, where the delta G is the difference between energy between constants. So what does that mean? It means that first, initially, when I just have my protein and no ligand, then I have my free energy landscape, and the probability of its state of the system is proportional to the disposal factor where the free energy I put in the conformation of free energy only. Now, when I add the ligand, I link the conformational equilibrium to the binding equilibrium, right? So it means that I multiply the conformational equilibrium constant by the binding equilibrium constant, which is equivalent to simply adding the two free energies. So now, if I imagine my free energy landscape, now it's changed because I've added this binding free energy. So the location of the lowest free energy might have changed here according to the binding free energy. In fact, I could do the same thing with the binding free energy that I've done with the conformational free energy. I could plot it over the whole conformational space of the molecule. And this gives me the binding affinity landscape. So I have a free energy landscape. Here I'm plotting, let's imagine here I have my conformational space. I have my free energy landscape. I have my binding free energy landscape. And when the water molecule is bound, the free energy landscape is now the sum of those two. I could call it the ligand perturbed free energy landscape. So now in the Locke-Key model, we assume that before the ligand bound, we just have one conformation. So that meant we have a free energy landscape with a very deep, narrow minimum, single deep, narrow minimum. And if the then let's say the ligand already fits perfectly with the shape, so it will only bind to that one conformation. So the binding free energy landscape looks exactly like the original free energy. So Locke-Key requires a very deep, narrow minimum in both the free energy landscape and the binding energy landscape in the same place. So in other words, the fit is between the conformational free energy landscape and the binding affinity landscape. Okay? Everyone agree with that? Any questions? So then let's think about the other models we talked about, like conformation capture. So remember that in conformation capture, we assumed that before the ligand bound, you had a flexible binding site, active site. That means that your free energy minimum was broad and shallow. But remember, when the ligand bound, it fixed the conformation in one place. So that means that the binding affinity landscape was narrow and deep. And then when the ligand's bound, you have a narrow and deep affinity. So you just have one stable structure. So the conformation capture results from a deep, narrow minimum in the binding affinity landscape and a broad, shallow minimum in the free energy landscape. And then remember, the other model we had was induced fit. So induced fit, remember what Hoshland said. Hoshland said that the fit occurs only after the ligand is bound. That means that the ligand must be able to bind to the original structure. But once it's bound to the original structure, it's converting to another structure. So that means it must bind with even better affinity to the new point in the conformational landscape. So our binding landscape has, it's going down, it has a minimum at the original conformation, but it has a lower minimum in the new down conformation. So induced fit results in the overlap of the binding landscape with the original preemptive landscape, but with a minimum of the position. And that actually, in general, would seem like the most likely scenario, although it will vary from one system to another, depending on the shapes of the two energy landscapes. Okay, so to summarize, liquid perturbed preemptive landscape is just the sum of the other two. So the probability, I should add that this is for the fully bound state. Okay, so we're assuming the fully bound state is not equilibrium. And if we could predict, so if we knew what the binding affinity landscape would look like, and the preemptive landscape would look like, we could predict that we could engineer the biomolecular conformation. Remember, that's what we said. We still haven't figured out to do it, even with artificial intelligence. But in order to be able to do so, in order to get this binding landscape, measure it, we would have to isolate the fixed representative components. And in general, in our experiments, when we measure a binding, then we measure it in a system where we have ensemble molecules of solution. And what we're measuring is an average of all those molecules, average of all the molecules. So we don't get the binding end for each specific compound. So ideally, we could fix the conformer and then do the binding. But it's very difficult to fix the protein secondary or tertiary structure. For example, you might fiddle with the sequence or truncate it or something in some way that you'd be able to fix it just to form one conformation. But that's very difficult with proteins. But with nucleic acids, at least with RNA, at least with what we call the secondary structure, the space theory, it's actually possible to make this prediction and then fix the conformers. And that's simply because of what we learned before, the simple roots of the base theory. So what's the ideal model system to test this approach? To argue it's the riboswitch. A riboswitch is a type of RNA. It occurs in what's called the 5' UTR. 5' UTR is 5' untranslated region. So it's a region of the gene that's upstream from the part of the gene that actually expresses proteins. So it doesn't express, it doesn't code for any protein. But it forms